Okay, so welcome everyone, and thank you very much for joining us today. Today we've got an exciting seminar from Professor Andy Borrell, who is known to many of you, but for some may not be known. And Andy's going to be talking about from RHT genes in wheat to PIN genes in sorghum, a life in crop science. Next slide, please. For those who don't know me, my name's Ian Godwin. I'm the Director of the Centre for Crop Science at the Queensland Alliance for Agriculture and Food Innovation here at UQ. Next slide, please. And the University of Queensland acknowledges the traditional owners and their custodianship of the lands on which we meet. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. Next slide, please. Okay, so just to remind people, so our seminar we'll be ending the seminar by 1 p.m. So Andy's going to probably talk for 45 minutes, and we'll have some time for questions. If you do have a question, rather than use the chat function, there is down the bottom a Q&A tab. Please use the Q&A tab, and then we'll be able to ask. I will then ask your question. And if you identify yourself by your name, I'll be able to say who that is from as well. So just remember the Q&A tab. Okay, next slide, please. Okay, so soon we'll be handing over to Andy. Andy is a professorial research fellow in the Centre for Crop Science. So Andy's actually an original product from Melbourne. He went through university in Melbourne to start his brilliant career. I did have a bit of a sneak preview with Andy of his talk, and I remembered back at a time when Andy and I would have been undergraduate students, and we'd have these old guys showing us faded slides of things with photographs that were taken probably in the 1950s and things like that. And yeah, it turns out Andy and I have turned into those guys. But it means we've got lots of good stories to tell, right, Andy? I will say that Andy, prior to working on sorghum, had actually done some work on rice, and then he decided that having moved from something that grew in water, he was going to move to something that grew where there was hardly any water, and that was dryland sorghum. So he was a founding member of the sorghum group, which became the Oz Sorghum Group. We used to meet at Bribie every year, and they became known as the Bribie meetings. And the first meeting was in 1993, and Andy and I were there along with people like Graham Hammer, etc. Andy joined the University of Queensland as part of QAFI in 2010, so he was one of the founding members of QAFI. As you'll see from his presentation, he's led a really exciting life of international scientific research and discovery, and he's focused predominantly on drought adaptation in cereals. I will also just let you know that Andy's a Churchill Fellow, and he's based at a research facility, and for a number of years he was actually the centre leader at Hermitage. So without further ado, welcome Andy, and over to you.
Thanks, Ian. I too acknowledge the traditional owners and their custodianship of the lands on which we meet, and pay my respect to their ancestors and their descendants. Well, good afternoon, everyone. I'm going to give an overview of research that I've been involved in over the past 40 years, and I'm going to focus on my early to mid-career, and hopefully there'll be some learnings for the younger scientists. I learned fairly early on in my career that you don't achieve much by yourself in science. You do good research because you work with good people in good teams. And so this is a story of team research. And to speak on the RHT genes in wheat, to PIN genes in sorghum, a life in crop science. And I want to begin with the early years. Now, I grew up in Melbourne, and when I was younger, and I don't know whether this has happened to you as well, but you go out with your parents and the other adults there and say, well, Andrew, what do you want to be when you grow up? And these are some of my answers. When I was 11, I dreamed of being an astronaut. At 12, I wanted to be a rocket scientist. At 14, I was going to be a geologist. At 15, a biologist. At 16, a medical doctor. And at 17, an agricultural scientist. So you can see there's a theme of science, but all different variations of that theme. My parents had a real influence on me as well. So my mother, Delma Joan Borrell, had a real interest in projects in developing countries, in agriculture and in medicine and health. My father, Murray Howard Borrell, he studied organic chemistry when he was younger at RMIT, worked briefly for CSIRO. He spent the second half of his life as a botanic artist living in the rainforest up in North Queensland. So they both had a profound impact, I think, on what I ended up doing. This is a photo of me when I would have been about 10, and I discovered that my father had all this amazing glassware stored under the stairs in our house in Melbourne. And I would come home from school and do experiments, and it was just such a tremendous setup and a lot of fun. I remember one day when I was about 10 years old, so this is in 1969, we were sent home from primary school to go and watch this amazing event where Neil Armstrong climbed down off the ladder onto the moon, and on our black and white television at the very bottom it said live from the moon. So science is inspirational, and I thought at that time that if science and technology can allow us to do that, then I want to be a scientist. My father gave me this book, The Living World of Science in Colour, probably when I was 9, and it had stories about all the great scientists from the Renaissance through to the stage when I read it, and it was very inspirational. At that time too, I used to write these letters to NASA in the USA, and it goes something like this: Dear NASA, I'm 10 years old. My name's Andrew. I want to be an astronaut when I grow up, so please send me some material about your astronaut training program and anything you can about astronomy. Well, to their credit, NASA actually wrote back to me, and they'd send these amazing packages full of wonderful information about space and science and technology, and it was just great. The lesson here is that you're never too young to be a scientist. Why crop science? Well, my family moved from Melbourne to Coffs Harbour on the northern New South Wales coast when I was ready to go and do year 11 and 12. So I went to Aurora High School. I wanted to do physics, chemistry, maths, and biology, but you couldn't do biology on the same stream as chemistry and physics and maths, so I had to do agriculture. But I soon discovered that I loved it because it was this combination of discovery science with practical application to dealing with issues like food security. So that was my introduction. I then decided that I would study agricultural science and go back to Melbourne because that's where I'd grown up. So I did a Bachelor of Agricultural Science at the University of Melbourne. And in my 4th year, I specialised in animal plant genetics and in animal plant physiology. Now, which direction to go in? When I finished my bachelor degree, different companies would come onto campus to say, well, you know, how would you like to work in this area? The ANZ Bank was one of these. And I went for an interview with them because they wanted agricultural scientists who understood something about commodities, and I had studied economics as well. So they offered me a job. And then I had to think, well, what do I do? Because I'd also been offered a scholarship to do a master's degree at Melbourne Uni. So it was international banking or international agriculture. And for me, the lesson here was to follow your heart because the agriculture area was something that I really wanted to go into. The 1980s. I had a scholarship to look at the RHT Green Revolution genes in wheat at Melbourne Uni. And as part of that deal, they wanted me to spend 1982 to understudy Dr. Albert Pugsley. And Dr. Pugsley was at the time one of the most well-known wheat breeders in Australia. He was 72 years old at this stage. He was forthright, some would say cantankerous, and he developed these set of near isogenic lines with the RHT genes in a triple dirt background, and they were ready just for physiological dissection. In the first weeks that I was working with Dr. Pugsley, he walked into my little cubicle there and he slammed a book down on the desk and then just said, "Read this," and then he marched out again. Well, it was Norman Borlaug's biography, and it really was a great story. Why study the RHT genes in wheat? Well, Dr. Pugsley, there's a photograph of him here with Dr. Borlaug in 1968 at Wagga Wagga in Australia. And Dr. Borlaug was developing the RHT genes, Green Revolution genes, shorter plant stature, higher harvest index, and he was doing that on an international scale. Dr. Pugsley was getting material sent to him from Dr. Borlaug with the RHT genes, and he was working at the Australian level and incorporating that. So you can see they were about the same vintage, good friends, collaborators, and it really was a tale of two wheat breeders. Now, Dr. Pugsley had a few favourite things, and one of them was serendipity. So we would go out into the wheat fields at the Melbourne Uni Research Station at Mount Derrimut, and this is how I'll define serendipity. Cambridge Dictionary says the fact of finding interesting or valuable things by chance. And dictionary.com says an aptitude for making desirable discoveries by accident. So this was one of Dr. Pugsley's favourite things. We'd walk through a paddock, he'd bend down, look at a wheat plant and say, well, what's that on the leaf, Andy? Or why is it that colour? Why is that leaf curled there? So he was always asking questions. And the lesson here is to look for the unexpected. There were some quotable quotes from Albert. One of them was, "Become a wheat breeder, Andy." And he said that to me on more than one occasion. He wasn't very impressed that I wanted to be a physiologist. And then one day he says this to me, "All physiologists ever do is tell breeders why they got high yield 10 years later." So these were some of the statements from Albert about be a breeder, Andy, and maybe not a physiologist. But after I'd done the studies on the physiological dissection of these RHT genes, he was quite pleased that he had a greater understanding of how they worked. So in the end, I think he was quite okay about it. The lesson is here, be teachable. If you are lucky enough to be surrounded by some people that might be older and wiser than you, as I was with Dr. Pugsley, then that's a great thing and you can learn a lot. I did get the chance to meet Dr. Borlaug at some stage later on. This is a photo with him in 2002. And I think Dr. Pugsley would have been pleased that we got together. The University of Melbourne was an amazing place at this time, and the Dowling lab in the early 80s was phenomenal. Mike Dowling was a tour de force in plant science at that time, and particularly in developing biotechnology. And here's a list of some of the postgrads that were there with me at the time in the Dowling lab, and they all went on to have quite remarkable careers. And the lesson here is that early friendships are important, so value them. And some of the ones, whether you're an undergrad, a postgrad, a postdoc, the friendships that you develop in your early years, they may be friendships over a 40-year period, like some of them have been for me. This is a photo of a young Evans Laguda and myself at our first scientific conference in Perth. And this is an example of a friendship that Evans and I have continued over the past 40 years. We did get round to publishing this work on the RHT genes on the physiological basis of their operation. And these 3 papers in Annals of Botany are special because they're the first ones scientific papers that I published, and they're always special. When I was about to finish my master's degree, I made a decision to go to India to have a look for a few months because I wanted to look at the impact of the RHT genes on food security and to see the extent to which they were making a difference. So I set off there just before Christmas in 1984, had about a month there. Then my new wife, Carolyn, came and joined me. And we spent time with a team of agriculturalists, educators, medical doctors in villages, and just looking at the impact of the Green Revolution. And I became convinced that it was a really worthwhile thing to put time into. So the lesson here is just make things happen. Melbourne University has had more to offer at the time than just intellectual pursuit. And I think it's great to be able to explore the wider life at the university. And for me, that was being involved with the Melbourne University Athletics Club, MUAC, as a 400 and 1500 metre runner, and also the Melbourne University Mountaineering Club, the MUMC, where we rock climb at Mount Arapiles and ice climb at Mount Feathertop. And I've made some great friendships in those years and explored some different parts of who I was. So the lesson here is that a university education is more than just getting a degree. When I finished at Melbourne Uni, I was going to go and do a PhD either in Australia or the UK or the US. A job came up in air with the Queensland Government as a rice agronomist. And I thought, well, I'll apply for that position and just see what happens. And I did apply and I got offered the job, and I said, okay, I'll take it as long as you allow me to do a PhD straight away. And they did. So Carolyn and I headed up to North Queensland, and the lesson here is embrace the unknown, because it was completely different in terms of particularly the climate, the culture, the lifestyle, everything. And it was an amazing experience. We faced cyclones. In fact, in 1988, 89 and 90, we had Cyclone Charlie, Ivu and Joy. We used to track them those days in the local newspaper. And you can see here Cyclone Charlie in 1988 coming straight into the coast. Cyclone Ivu in 89 was a category 4. The eye went over air, and there was a lot of damage and quite an experience. My PhD studies at UQ were about novel water-saving strategies in rice. And this was driven by the rice industry at the time, because 40% of their total variable costs went in paying for water. So they wanted to see if we could develop a system for growing it with less water. So with Shufukai and Alan Garside, we worked on this technique called saturated soil culture and reduced the water from 12 megalitres per hectare back to about 8 with similar grain yields. And we published that work in Field Crops Research and some other nitrogen papers as well. This is a photo of me with Lindsay Hall, who then was chairman of the North Queensland Rice Growers Association. And the point here is that when you worked as an agronomist with the government, pretty well every week we spent time in the field with farmers. And it was such a great way to learn, and over time to provide assistance where we could as well. So the lesson here is to build partnerships with industry if you want to have impact. The 1990s and beyond. Well, when I finished in my PhD in my time in North Queensland, had to work out what was next. And Shu Fukai, who was my PhD supervisor, had an ACR position going. He had a project in Thailand. He said, well, Andy, why don't you apply for that? Got a postdoc position. I applied and got that. We're looking for a house in Brisbane. We're about to start that. And Shu rang up and said, oh, sorry, but ACR have delayed the funding for 1 year. So it's not going to happen at the moment. So I switched courses pretty quickly, and the government said, well, we've got a position at Hermitage as a crop physiologist. Would you be interested in that? And I went down and had a look, and it looked like a great opportunity. So that's where we ended up. And the lesson here is whichever fork in the road you take, you can make it work, because we have this freedom to choose how we respond to any given situation, which is an extraordinary thing. So 2 threads were developing in my career at this stage. Physiological section of stay green in sorghum was the first thread, and the other one was novel water saving strategies in rice. And I want to focus here on the stay green bit to begin with. And I'm going to switch to a theme approach here. We've been going through chronologically. Now I just want to have a look at a couple of themes. So the physiological section of stay green in sorghum, which went from 1993 through until the present. So what is stay green? Delayed foliar senescence, known as stay green, is a drought adaptation trait that enhances crop productivity in the field when water is limiting after flowering. There were 2, I'll call them fathers of stay green. And this is Dr. Darryl Rosenauer from Texas A&M in the US, and Dr. Bob Hensel from the Queensland Government in Australia. And both of these guys have passed away now. They were amazing sorghum breeders. They were great friends, colleagues, and they both noticed in the 1970s, walking through their sorghum fields, that under a severe drought, while most plants were dead and leaves were dead, in some cases the leaves stayed green. And they noticed a positive correlation between stay green and between grain yield. Now, when I started at Hermitage in 1993, Bob would walk in and out of my room, and in the first week he said, well, I know there's this positive correlation between stay green and yield, but I don't know how it works. You're a physiologist. Your job is to go away and find out the physiological basis of how it works. So that was the challenge that Bob threw out. In the early days, we grew sorghum under huge sheets of black plastic in the field to exclude rainfall. And we then applied water under the plastic via drip irrigation to create 3 water regimes. And this is a paper that I published here with Graham Hammer and with Bob Hensel. And you can see here that there's 3 water regimes, no water deficit, post-flowering and terminal water deficit, and 3 female parents bearing in stay green were crossed with 3 male parents bearing in stay green. And you can see the graph on the left-hand side here. On the x-axis, it's relative rate of leaf senescence, and on the y-axis, it's grain growth rate, and a negative correlation. And you can see there that the stay green types had a low rate of leaf senescence and a high grain growth rate. CSIRO were also working on sorghum at this time, and they developed a set of recombinant inbred lines, and they asked if I'd go over to India, to ICRISAT, 2 seasons, 95-96 and the 96-97 season, and phenotype their recombinant inbred lines in the Rabi season where it was extremely dry. So I headed over for these 2 years, both at flowering and at maturity. And it was one of those aha moments for me, because they had about 160 lines. It was a severe drought. And you'd walk through the field and most of the plants were dead, most of the leaves were dead, but occasionally you come upon a plot where the leaves were green, stems were green, and the panicles were significantly larger. And there's a quote that I wrote back in 1996. And for me, the adventure of tracking the stay green phenomenon had just begun. We published this work with Fran Bittinger and with Sunita. And the left-hand side, you can see this is greenleaf area, big grain fill, positively correlated with grain yield, and the relative rate of leaf senescence on the x-axis, negatively correlated with grain size. And this was exciting because in a bigger population, we were getting some understanding of how the trait worked. There was a pivotal meeting in 1999 that was run by the Rockefeller Foundation, and they brought together scientists that were working in 4 crops, the big 3 crops of maize and wheat and rice, but also sorghum. And the aim was to develop some research plans that had commonalities between the different crops. They did end up funding 2 of the big crops, but they did not fund sorghum. However, because we had a wonderful group of sorghum scientists there from around the world, we decided that we'd do it anyway. So the lesson here is if you don't succeed initially, keep on trying. We didn't get funded here, but we did go away and we decided that a group from Australia and the US and India should pursue the stay green trait and that we'd go and get our own funding, and we did. The funding for us in Australia came from the Grains Research and Development Corporation, and in the US they had some funding through Texas A&M and also through the National Science Foundation. This is a photo here of David Jordan and I in 2000. We went across to first work with John Mullett and with Bob and Trish Klein, and Karen Harris as a PhD student there. And you can see Norman Borlaug looking over John Mullett's shoulder, because at the time, John was the director of the Norman Borlaug Centre for Southern Crop Improvement at Texas A&M. Other collaborators here were Henry Wen from Texas Tech and Darryl Rosenau from Texas A&M. And Henry Wen and Darryl Rosenau developed 34 near isogenic lines for mills for stay green. We took those, or Darryl sent those 34 lines to us, and we grew them under 4 environments: a water-limited treatment with high and low density, and an irrigated control with high and low density. And we did a detailed physiology and fine mapping study. We published this work initially characterizing the 34 stay green regions and some preliminary work about how the trait worked physiologically. And then we did some summarize the physiological understanding. And if you introgress a single stay green QTL region in, you get a decrease in water demand, and that decrease in water demand is due to a reduction in tillering and a reduction in the size of the upper leaves, and it results in a smaller plant size at flowering. But also the trait causes an increase in water supply, and that's partly due to shifting of water from pre-anthesis to post-anthesis, but also because of a narrow root angle which sends roots deeper into the soil to access water at depth. And these things together result in an increase in grain yield. And we published that work in the New Phytologist and in JXB, and looking at stay green's association with canopy development, leaf anatomy, root growth, and water uptake. We did some translational research as well, rolling out of stay green technologies in India and in Africa. And it took us a little while to get our partnership going with India. Remember back at the 1999 meeting with the Rockefeller Foundation that we had wanted to do that? Well, it began with an ACR project that went for 10 years from 2008 to 2018. And it was based in West Central India in 4 states there. And you can see here that the leader of this project was Vincent Des, Adicrosat, And you can see some Australian scientists involved, Graham Hammer, Greg McLean, and myself, and some of the national program in India, Talwar, and others. If you travel through this part of India and you stop and get a meal, then commonly you'll have something like this. It's a roti jawa, a flatbread made out of sorghum. You might have a yogurt, and that can be from the dairy cattle that feed on sorghum plants in that area. So the project we had was looking at dual-purpose sorghum, particularly on grain sorghums, and looking at the integration of various stay-green QTL into elite material. Now, this is S35, which is an elite Indian material. It doesn't look very elite here because under drought it crashed and burned. But if you integrate the single stay-green QTL into that, stay-green 1, then you get a plant like this, where you've got green leaves, green stem, and a high grain yield. Now, this is where serendipity or the unexpected comes into it, because we found here that the stay-green QTLs integrated into S35, and these plants produced a higher quality stem and leaf material. And this increased the palatability, digestibility, metabolizable energy, and milk yield of cattle. So about a 10% advantage in both grain yield and stover yield by integrating stay-green QTLs. And here's Talwar showing us through some of the trials where these varieties were being rolled out across India. And Vilas Tanapi was telling me recently that 2 of these lines have gone out and at the moment are being released through the All India program commercially and available to grain growers in these parts of India. So that's a great outcome. And then we published the work on these different recombinant inbred lines and physiological studies that we undertaken and the introgressions, and also looking at the food and fodder traits. We did some field testing of stay-green in Africa as well between 2009 and 2015. And this was work that I was doing with David Jordan in West Africa and in East Africa. David developed some Malian by Australian germplasm, and this was tested and evaluated in 2013-14 at 2 sites in 6 African countries. And here's an example of some data from Mazangalini, which is in Kenya. And you can see a green leaf score on the x-axis against grain size on the y, and a positive correlation there. And discussions with sorghum breeders from Mali and Burkina Faso and Kenya highlighted that this Malian times Australian germplasm was broadly adapted across sub-Saharan Africa. Now, this thread on stay-green, I want to finish by saying something about the pin genes in sorghum and the molecular basis of stay-green. Now, this was work that was done with my sorghum buddies at Hermitage, Emma, Alan, Barbara, and David. And it's a team with a combination of molecular biologists and plant breeders and physiologists and geneticists. And you'll see Alan Cruikshank here as well. So Alan passed away last year and we still miss him terribly. But it was a great team that we had for doing this work. Now, simultaneously to this, and more recently, we had the ARC Discovery Project between UQ, CSIRO, and Justice Liebig University that was working on this aspect of the pin genes in sorghum as well. Albert Wong, Yasmin Lam were PhD students on this. Karen Maisel was a postdoc. Ian Godwin and Lee Hickey were CIs on this project with me as well. And some of the results from this were why pin genes, or members of the pin family of oxen efflux carriers, often appear within stay-green QTL. And 9 of the 11 genes from the pin family in sorghum co-locate with Stage-Green QTL. We're particularly interested in the Stage-Green 1 region, Stage-Green 2 region, and David had been selecting for the Stage-Green 3B region within the program at Hermitage as well. We developed some transgenics. So this is work done by Ian Godwin and Lou. And you can see here, Texas 430, if SB-PIN2 is down-regulated there, you get a unicolm and a small leaf area. Upregulation, overexpression of SB-PIN2 gives a high tillering phenotype with a high green leaf area. And we published this work in JXB, looking at genetic modification of PIN genes and how it induces causal mechanisms of Stage-Green. And Albert did more of a review paper on this for Annals of Botany Plants. Alongside the years of work that we've done in Stage-Green in sorghum, we also worked from 2004 through to the present on Stage-Green wheat. And this was a team that was largely headed up by Jack Christopher, with significant input from others, including Corinne Chenu. So the second thread that I was working on was novel water-saving strategies in rice in Asia, and pioneering these strategies. I did a Churchill Fellowship in 1988, and I spent some time at the International Rice Research Institute in the Philippines, and at 10 institutions in Japan, and then at a number of research institutions in the southern United States. And I focused on water-saving strategies in rice and also nitrogen dynamics. And coming back from that, I got into my PhD studies, and from an earlier slide, you'll recall that we were looking at saturated soil culture. I had a friend, Don Van Kooten, who came through AIR when I was doing these studies, and he was a UQ graduate as well and working in West Timor. And he said, "Oh, Andy, come over, this sort of technology, it'll work in the villages in West Timor." So for a 6-year period from 1993 to 1998, I went over with Don in my holidays and just funding it ourselves. And we did these experiments at the village level with raised bed cropping technologies, going from a monoculture through to a more diverse system. And the lesson here is to be proactive. I'm proud of this paper that we published together on enhancing food security in semi-arid eastern Indonesia through permanent raised bed cropping. Because it contained 10 components of a system, looking at all different things from compatibility of raised beds to upland cropping to weed control, erosion, all sorts of things. And then the lesson here is to publish good science and funding will follow. So we did the experiments, we published it, and then other organisations, ACR and the Queensland Government, World Vision International, began to fund this work as well. I'd been working in West Timor through until the end of 1998, and then in 1999, an independence, a referendum was held in East Timor on the 30th of August, and the East Timorese people voted for independence, leading to mass violence before the UN was able to restore peace. And the Queensland Government were very keen to be involved in nation building in East Timor, and they asked myself and my colleague Rob Kelly, who'd also been working with us in West Timor, if we would be interested in going over and training some of the East Timorese agronomists. So we did this in 2000. We ran 3 workshops, and then again in 2002 we ran another 3 on rice-based cropping systems and running field experiments and other things. And it was an extraordinary experience to work with these very courageous and amazing people. Then, following on from that, there was an opportunity for Don and I to be consultants on an ACR project in Lombok that went from 2001 to 2008. So the lesson here is that one opportunity leads to another. I began working with Don in West Timor, and self-funded work, and then East Timor was Queensland Government, World Vision International, and the Crawford Fund, and then Lombok with ACR funding, Laos and Cambodia with ACR, and then more recently working in Vietnam, and then India with the Norwegian government. One of the things that happened, it didn't matter whether it was in Indonesia or in India or in Africa, was the development of partnerships. And this was very much about coming in and meeting with the locals, the local farmers, the local scientists, local government officials, and listening to what they had to say. They knew more about the soil and about the agriculture and about the challenges that they faced than we did. And that was a great experience together to sit down with them and to work out, what they needed. That was basically us asking, well, how can we help? So the lesson for me here was very much learning to listen. And then the work in Lombok, we are able to look at raised beds versus flooded and showed that you could produce a similar grain yield with both systems. The water requirement in raised beds was half that of flooded, and the water use efficiency was almost double. And then from 2013 to 16, I worked in Vietnam with the Norwegian Institute for Bioeconomy Research on climate change and impacts on rice production in Vietnam. Now, I've talked about two threads, but there were a couple of other highlights that happened along the way, and I just want to give a snapshot of a few of those. There was an explosion of new opportunities that coincided with the build-up and launch of coffee in 2010. And the lesson here is to step out of your comfort zone and embrace change, because it was a big change from the government going into a new institute, but what an amazing journey it's been. I worked for a period of about 14 years with Jack Christopher and Troy Fredericks on in-head frost tolerance in wheat and barley. And we even led the national program for a 5-year period and made some progress there. There's a GRDC lodging resistance in sorghum project that I'm involved with David Jordan and Giedicke, addressing one of the biggest challenges in the grains industry to develop lodging resistance. I have involved in a small project in the ARC COE for Plant Success in Nature and Agriculture, working with a PhD student, Chaitanya, and with Alex Wu and Eric Van Oostrom. And it's a UQ-UTAS partnership linking stomatal architecture and hydraulic conductance. With the running of Hermitage for a long time now, we had the centenary back in 1997. To commemorate that, we had a Hermitage Schools plant science competition, which we began at the time, and it's been running ever since. And you can see here Kerry Ruby, who has been the lead running this, and more than 100,000 students around the country have been involved in this, and we've been able to introduce them to science, and particularly agricultural science. And Costa from Gardening Australia has been our guest on 2 of these occasions. One of the great privileges for me has been leading Hermitage Research Facility for a period of about 14 years. I've handed that over a few years ago to Dr. Barbara George-Jagley, who's doing a great job. And the lesson for me here is that leadership is a privilege. Get good mentors. And I had some great mentors in Paul Johnson, Bob Hensel, and Joe Baker, and they helped me a lot. Where to from here? Well, I'm going to be seeing less of the office because I'll be retiring from UQ at the end of the year, but I will be spending some time continuing in science with some PhD students and being involved in some science projects. I'm going to do some other things as well, though, and one of them is investing in grandchildren. Carolyn and I have 7 grandchildren, and grandparenting is part of our legacy, and this is something that I'm looking forward to a lot. One of my pastimes that I really enjoy is surfing, and there's 2 of my sons, Lee and Chris, and we get out there on the boards together and with my wife Carolyn, and that's Greenmount, which is one of my favorite beaches in the world, and maybe I'll see you out there. Mentoring the next generation of scientists, what a privilege. And this is a photo of some of the IRTG group, the collaboration we have with Germany, and it's just such a privilege to put time into the next generation. I've also enjoyed expeditions through my life, and there's 3 ecosystems that I'm particularly interested in: alpine, marine, and rainforest. And I'm hoping that I might be able to be involved in some of these in future as well. So that's all from me. And with that, I'll hand it back to Ian. So thanks, Ian.
Thanks, Andy. That was great. Look, I couldn't help but notice that the 3 ecosystems you're totally interested in are as far removed from semi-arid tropics as you can possibly get. I wonder if that's a reaction. Maybe you just want to keep work and play separate. I wanted to kickstart the questions by asking you a little bit more about Stay Green. So can you give us a bit of an idea as to how and where has selection for Stay Green in sorghum become a normal part of plant breeding? Is it something that is contributed just in Australia, or is it a worldwide phenomenon?
Yeah, well, it certainly had a strong push in Australia with the work initially of Bob Hensel, and then David Jordan's been following this trait very strongly as well, and working with grain growers. And there's a strong collaboration between DAF and UQ and with the private sector, which David's been pushing hard to. And so there's some real wins, and it would be the major drought adaptation mechanism that's used by the private companies in Australia at the moment. And it's the main mechanism that the grain growers have in their fields in Australia. It's also been, as you saw, the work that's being done in India by groups like ICRISAT, and by the Indian Institute for Millet Research, that it's one of the drought adaptation mechanisms that they are introgressing into their material now. And also in India, or in Africa, it seems from talking to their breeders that the Stay Green trait is one of the mechanisms that seems to work well across sub-Saharan Africa. So essentially anywhere where you've got severe post-anthesis droughts, and it can be parts of China, India, through the United States, Australia, parts of Africa, and even regions where we have not thought that would be the case. It's becoming more important with climate change as well.
Okay, thanks. Well, following on the Stay Green theme, we've got a question here from Anthony Van Heerwaarden. He says, given the improved fodder quality at the end of the season in the Indian experience, were any of the Stay Green QTLs associated with increased nitrogen uptake?
Yes, they were, and in fact, it was interestingly, nitrogen dynamics were one of the things that we followed with our earlier studies. And we found that because the Stay Green types were taking up more water during the grain filling period, through mass flow, they're also taking up more nitrogen. And so that was a mechanism of increased nitrogen uptake. And then with the near isogenic lines in our rainout studies, we also measured that directly and found that the nils with the stay grain introgressions took up more nitrogen during the grain filling period.
Okay, thanks. And now we've got a question from Sudhir Yadav, who says, congratulations on a successful journey and contribution to science. But his real question is, do you think breeders' perception of physiologists have changed now, or do they still believe physiologists take 10 years to address questions breeders already know?
Yeah, that's a great question. I can remember being introduced about 20 years ago at a conference in Texas as a physiologist and as a near extinct species because I was a physiologist. And so at that time, there probably weren't too many physiologists in crop improvement teams. But I think increasingly that crop physiologists have become more a part of crop improvement teams. So with plant breeders and people doing quantitative genetics and molecular biology and genomics, the physiology, because it helps to understand gene action and gene networks, then I think it's becoming an important part, and the whole modeling part as well involves a strong physiological framework. So yes, I think it probably has changed, and yeah, it'd be interesting to see what Dr. Pugsley thinks of that now.
Definitely, definitely. Our next question is actually from a visitor from Germany, from HHU in Dusseldorf, Gertz Hensel. He's said that the high tillering phenotype is related to strigolactone biosynthesis and degradation. Have you looked at strigolactone in the PIN2 overexpressors?
Yeah, not directly. We've been looking at the PIN genes, which are auxin efflux carriers. And so at the moment, it's been, more looking at it in terms of auxin. But yes, some of the role of some of the other hormones would be very interesting to pursue.
Okay, thanks. Our next question is from someone called Anonymous. Said it's very inspirational seeing that genomics is a big theme in crop science. Do you need to be good at molecular biology to pursue crop science? And I promise it wasn't me who asked that question.
Yeah, the one thing I've learned is that if you're a specialist in one area, which I guess most of us are, so for me as a crop physiologist, I realized that in my career that I had to try and get up to speed enough in other disciplines to at least be able to hold an intelligent conversation with the plant breeders and the molecular biologists and those doing modeling and biochemists. So we work in multidisciplinary teams, and as I said at the start, we only achieve anything because we work in these amazing teams. So having some knowledge of the other areas that you work in is important. So yes, the molecular biology it's an important component of the crop space. And I think having some knowledge of the plant breeding, quantitative genetics, molecular biology, physiology, modeling, and these days of bioinformatics and areas like robotics, having some understanding of those, even if you're a specialist in one particular area, would be really helpful.
Yeah, great answer. Next question is from Phil Banks, who says, if you had your career over again, is there anything you would do differently?
Yeah, that's a good question too. As I said, I've really enjoyed it, but I also made the point that there are forks in the road where you decide to do things, and all of us face this, and you go one way or you go the other. And what I would say is that in a sense, it doesn't really matter which way you go, because you're gonna make it work. Because you've got the capacity to respond to a situation and to drive it in that direction. So no, I'm really happy with the direction that I've taken, but I also understand that I could have taken alternative routes, and they could have been very fulfilling as well.
Yep, that's very true. I mean, after all, you could have introduced the cane toad to Queensland to be a biocontrol pest, and then you'd be famous for all the wrong reasons. We've got another question from an anonymous attendee who says, seems like you've been part of a lot of different research topics and projects, and I assume there's been many more that you didn't present. And really we're saying, how did you decide what was worthwhile, especially early on in your career? So it follows on a little bit from the previous question, I guess.
Yeah, I think in many cases it's the opportunities that are presented to you. So I wouldn't necessarily call it luck, but there are things, opportunities that come up. And so for me at the very start there, I was going to work on an ACO project with SHU. And because the funding was delayed by 12 months, then I ended up at Hermitage. So there's things like this that happen all the time in our lives. And I think it's opportunities that are out there for us. And then the challenge is, how do we respond to those opportunities? And sometimes I think, well, probably often, it involves stepping out of your comfort zone. So I think there's a degree of courage where, you've got an opportunity, you think, oh, what'll I do? And sometimes it takes a degree of courage, and you've got to step out of your comfort zone and just go for it.
Okay, and we've got time for one last question, and I'll tell you, it's a person who says, I would also add that physiology and modelling can help troubleshoot intervention consequences. It's from someone called Alex Wu, who you know well. And he says, he's really going to the punches now. He says, for example, stay green can reduce yield potential in a good environment. What would you think we could combine stay green with so it can improve performance of stay green in all environments?
Yeah, I think, well, there's a G by M component here too, and thinking about how stay green interacts with management practices as well. So I think there's this whole concept of the G by M by E, and for example, farmers with their planting densities, and they can modulate those in combination with their genetics to get a specific adaptation for their particular environment. So I think having it as a trait under genetic control and seeing the interaction of it with management and environment is a very exciting space.
Okay, thanks, Andy. Well, it's 1 o'clock, so I'm afraid we had a couple of questions we weren't able to get to, but that is great because it showed the engagement that you've been able to lead on your seminar. And thank you very much again for doing your seminar. I think we might have another slide or two to come. Is that correct? Just to remind people that this has been recorded, and you can actually go and watch the recording on our Science Seminars webpage. And do we have an announcement of an upcoming one at the end? Not yet. Okay. All right. Well, thank you very much. And thanks very much for the participants for joining us. And thanks so much, Andy, for taking us on a fantastic journey through a life that has contributed so much to our understanding of crop physiology and all the impacts you've made, not just in Australia, but around the world. So thanks again. Great stuff.